Hello, this is chapter 4. We're going to do parts 1 and 2 here. And so with that, of course, with every start of the chapter, we have the chapter objectives. So you can go through and read through them. We will basically be getting to I don't know, about here. We'll start comparing and describing the different types of cells for this part in this particular presentation. Keywords, a lot of vocabulary here, so do make sure that you know all these words. Introduction, so in this chapter, we're going to look at microorganisms. Ideally, we would have this lesson, or you would have or are doing it, depending on which semester you're taking, while we are doing this in lab. And so we are going to be looking at microscopes. We'll be learning how to use microscopes. We'll be learning what these organisms look like under the microscopes. We'll be looking at prokaryotic cells versus eukaryotic cells. Those are the two main types of cells. We will be looking at the components, what makes up a eukaryotic cell, and the organelles inside the eukaryotic cell, and the diversity of the organelles in eukaryotes. We keep talking about eukaryotes because eukaryotes have this compartmentalization with different organelles where prokaryotic cells do not, and that's what we're going to look at here. So first thing we're going to look at is, well, how do we study microorganisms? And so we have these fundamental domains. Remember, we talked about our domains in chapter 1. We have our 3 domains. We have bacteria and archaea that are classified as prokaryotes, which mean they do not have a nucleus. They also do not have membrane-bound organelles. So they do not have a Golgi, they don't have lysosomes, they don't have a rough and smooth ER. And some of you are probably going, what is she talking about? Because we will get to those organelles this chapter, I promise you. So they do not have these specialized structures called organelles like eukaryotic cells do. But I think you all know what a nucleus is. So prokaryotes do not have that nucleus. They don't have that protective layer around their DNA. Where eukarya, they are eukaryotic and they have a very clear nucleus. And what we're seeing here is we have a picture and we've got some bacterial cells there. We've got looks more like a toad that is part of eukarya, and a mimosa plant, which is part of eukarya. So basically all your single cell bacteria are part of domain bacteria and archaea, and that they are classified as prokaryotes. Everything else, all other life, is eukaryotic. Viruses are not considered living. And this is why we can't kill them, because they're not alive. We can't attack a function that they do, because they really don't do anything. They are just junk DNA. It's just like a computer virus, right? It's just junk DNA that messes up in the body in a way that it spreads that virus to other people, right? So viruses are not made up of cells. They do not metabolize, so they don't eat. They do not maintain homeostasis. They do not replicate on their own. They have to have a host cell. The host cell is making more copies of them. They also, there was one other thing I was going to say. I'm going through all my characteristics of life. They are a little bit organized. We can talk about that. Okay. So again, they don't meet all the characteristics of life. So they typically are just, it's a bunch of rogue genetic material, whether it's DNA or RNA, encompassed or encircled with protein. Right. So like I said, they do not display all the properties of life. So how we study these is by using microscopes, and the microscopes come in, there's all kinds of different types of microscopes. We will be working with what's called a compound light microscope. So what that means is that light has to pass through the specimen. We know cell sizes vary to very, very, very small to, well, an egg, right? An egg is one cell. So chicken eggs or an ostrich egg, right? They can be quite big. So the cells that are small enough that we cannot see with just our eyes, we will need to use some help. And so we're going to look at, and this would be our compound light microscope here on the left. And this is a big, huge electron microscope. Electron microscopes shoot electrons through the specimen for us to get an image. And so we have this thing called the cell theory, and cell theory states that all living organisms are made up of cells and that cells come from pre-existing cells. So we just can't spontaneously make a cell, which always gets us biologists in trouble because the students will always ask, well, where did the first cell come from? And then we have to say, well, we don't know. But we do have some ideas that are leading up to that concept of how the first cell could have formed. We're not going to get into that much detail in this class. So the second part is just comparing pro and eukaryotic cells. And so cells, again, they have 4 common characteristics. For one, whether it doesn't matter if they're pro or eukaryotic cell, they have a cell membrane or a plasma membrane. We'll find there's a lot of terms in biology that have multiple vocab words for it. So the cell membrane or the plasma membrane separates the interior of the cell from the external environment. They also both have cytoplasm, and that is, the cytoplasm is technically a space. All right, so it's the space inside. The space inside is filled with what's called the cytosol, which is the actual fluid, which is mostly water. And then we have DNA. So our bacteria have a genome that's one circular strand of DNA versus eukaryotic cells that have linear DNA. They have multiple chromosomes. And both have ribosomes. Ribosomes are an organelle that makes protein. Ribosomes are very unique organelles in that they do not have a membrane around them. They are made up of RNA, and we'll talk about RNA in a future chapter. So again, ribosomes make proteins. Cells fall into 2 categories. Like I said, prokaryotic cells, which are domains, bacteria and eukarya. And we can see our bacteria here. This is actually probably from Yellowstone, which if you ever have a chance to see that, those are hydrothermal vents. They are from volcanic activity. And so we have these are archaeans. They are thermal acidophiles. They like hot acidic environments, and we get some really pretty colors. That was one of my favorite things to look at last summer when I was in Yellowstone. I had lots and lots of pictures. And then we have domain eukarya. We can see all kinds of, and you'll look at this one. This is an onion. It looks like an onion cell. This is a paramecium. This is conidia, which we find in fungi. And then we got our toad from amphibia, class amphibia. So some characteristics of prokaryotic cells is, again, they lack a nucleus. Their DNA is found in a region called the nucleoid region. Like I said, DNA is usually organized in a circular chromosome. They lack membrane-bound organelles, and many prokaryotes have a cell wall. That cell wall is made up of a material called peptidoglycan. When you take microbiology, you'll learn that there are 2 types of cell walls, gram-positive and gram-negative. And so you'll study the difference between the 2. Prokaryotes are believed to be very similar to the original ancestral cell. So we started with prokaryotes and then became more complex into eukaryotic cells. Some characteristics of eukaryotic cells is that they do contain a nucleus. We can see in the picture here, we have all kinds of structures inside that cell. And so we have a bunch of organelles. Like I said earlier, the DNA is organized in the linear chromosomes, and eukaryotes can be further classified as animal, plant, protist, or fungi. Those are the 4 kingdoms there. Prokaryotic cells are smaller than eukaryotic cells. Surface area to volume ratios are more favorable for moving materials in and out. So what we're saying here is if you are smaller, we can get things out quicker, even though we can't move them faster. It's like we're changing its, remember that whole RT equals D. So if we change the distance, then it takes less time because we cannot change the rate. So if our distance goes up, our time goes up. If our distance goes down, the time goes down. And so the smaller the cell, the more efficient it can be at getting waste out and nutrients in. And so it's the surface area to volume ratio that limits the size of a cell. Now, multicellular organisms are going to have mechanisms to help with that transport of materials, and we have a whole chapter on that. And that allows those cells to get a little bit bigger. And so we can see here some comparisons. We can see bacteria are here versus our animal and plant cells are much bigger. So if you're not familiar with these sizes, a millimeter would be 10 to the negative third. That means 0.00 and then your number. And then you have your micron, so that would be your millimeter. Your micrometer, which is that is 10 to the negative 6. And your nanometer is 10 to the negative 9. So we're talking about things like the COVID virus. Those are measured in nanometers. So you think about point and then going out 9 places. Very, very small. So we can see that we have a virus up here, which is a flu virus, which is actually quite a bit bigger than the COVID virus. And then we can see a chicken egg, right? We can see a chicken egg with the visible eyes, and you can see naked eye can see pretty small here. And then we need a light microscope. And then when we get even smaller, we need an electron microscope to see it. So some of the things like I was talking about, factors limiting cell size is that surface area volume. And here's a picture showing this. And so as the cell gets bigger, it's a greater distance to get out from the nucleus. And so it gets harder to move things in and out fast enough for it to maintain homeostasis. So that surface area volume ratio is a very, very much so a limiting factor on how big a cell can get. Here's kind of a table showing this. So we can see like the side of a 1 by 1 by 1 cube. The surface area is 6, right? We would go 1 times 1, which would be 1. And then there's 6 sides of that cube. That gives us 6. And then if you know, so your surface area would be length times width. And then we have a cube. So since we have a cube, we have 6 sides. For volume, that's length times width times height. Right, so 1 times 1 times 1 is 1. That gives me a ratio of a 6 to 1. We want that ratio to be high. We want for a 2 by 2, we got 24 and 8. Notice our ratio is going down. And then with our 4 by 4, it's even smaller. And so it becomes less efficient at getting materials in and out fast enough. This is the main, like I said, this is the reason why cells are so small. So instead of us being made up of a few big cells, we're made up of trillions of little cells. And that is the end of 4142. If you have any questions, feel free to email me or write them down and bring them to class when we have lab. That would be a great place to ask questions in our in-person lab. All right, I hope this was helpful, and I will see you in the next video.